lecture. And let's jump in. So, during the last video lecture, we spoke a bit about sampling. We defined population and sample, and we talked about pitfalls that come up in sampling and things you shouldn't do. So you shouldn't, if you can avoid it, you should avoid voluntary response, convenient sampling, and find ways to control for this idea of sampling bias. So I'm not going to cover these in depth since they are all on the video lecture, but I would like to talk a bit more about good sampling practices that we can see and that we can do when we eventually start working with data, or when you start working with data IRL. So I'm going to call this section good sampling. Boom. And there's a picture. Ooh, not take a photo. Ah. This one right here. So on this picture, we have 3 types of sampling, and we're going to talk about them for a bit. So just a note on how to understand what I am attempting to visualize here. This first one is an example of a simple random sample, which we talked about in the video lecture, so I'm not going to define it here. And every single point on this rectangle is an element of our population, both the blue and the orange dots. And the orange dots are the subjects that we picked in our simple random sample. So remember, simple random sample is every single subject in a population has an equal probability, equal chance of being chosen, and we choose a subset of it just using sort of like a raffle type approach. So in this case, we raffled and we picked the orange dots, and I think there might be 18. So I think each of these pick 18 observations. So what you're seeing is a simple random sample of 18 observations. The next one we want to talk about, which is another example of a good type of sampling, is stratified sampling. So in stratified sampling, this is a divide and conquer sampling strategy. Where the population is divided into groups called stratum. So the reason we might want to do stratified sampling rather than a simple random sample is when we have subgroups of the population that we are interested in observing in the context of what other variables we are studying. And I think the best way to understand this is by means of an example. So suppose you wanted to know more about football. And I mean football in the international sense, so soccer salaries in different countries. So rather than draw a simple random sample, if you were to simple random sample, It's possible that one country could be overrepresented and another not represented at all. So instead of just putting all of the different football players into a pool and picking them at random, you would instead pick a random amount from every single team. So maybe 4 from Brazil, 4 from Portugal, etc. Right? And then within each of the teams, you would pick, in this case, my example, it looks like in each of the stratums, they picked 3 randomly. And within it is like a simple random sample. Every single member of the team has an equal chance of being chosen. So that's stratified sampling. The final one, which is actually a little more sophisticated, um, the nuances of this one we're not going to cover too much, but I wanted it to give at least give it an example so you'd be aware of it being an option when it comes to good sampling types. It's called clustered sampling. That is not how you spell sampling, Mer. And in this case, um, oh, how do I want to say it? It's the same as stratified, except we randomly select from which groups to sample. So as you can see in this little example, they randomly selected cluster 3, 4, and 8, and then within each of those clusters drew 6 observations. So it's like you initially randomly select the clusters, and then from each cluster you draw an equal amount of observations. And there are a number of reasons you might want to do this. I guess the example that I would think of is if you remember the county data from last lecture, there were what, 3,100, over 3,100 different counties. So maybe if you wanted to observe something county to county, you would randomly select counties and then select people within those counties that you randomly selected. So something to think about, but we aren't going to spend much time on this. So I have a poll. Now, I was telling the last class, I think I've decided I'm going to stick with the Zoom polls because I kind of like getting you all to read to me the results that I can't see. It's really refreshing to hear somebody's voice other than my own. So let's launch a poll. Or let's not. I'm now realizing I only made this poll for the first lecture. So I guess instead I'm gonna just ask you the question, and we're gonna see if anyone's willing to answer me. So let me pull it up. So you are interested in knowing the effects of a treatment on different age groups, so 10 to 19, 20 to 29, etc. And you plan to take a sample, then later break the sample into treatment and control groups. What type of sampling should you use? And I'm going to give you 5 options. And why don't you just, actually, since I want to hear from all of you, why don't you just post in the chat and we'll see what we have to say. So it's a simple random sample, voluntary response, convenient sample, stratified sample, or clustered sample. We are interested in knowing the effects of a treatment on different age groups.
Can you repeat the options again, please?
Of course. It is simple random sample, voluntary response, convenience sample, stratified sample, and clustered sample. Wow, look at all these responses. This is great. All right, so I'm seeing a ton of stratified, in fact, ubiquitously stratified. So very good, everyone. It is indeed stratified sample. I got some responses for clustered sample last class, which I guess you could make the case for clustered sample, but in this case, we aren't splitting the population into so many subgroups that it would be unreasonable for us to sample from every age group. So I would definitely go with stratified sample. You also wouldn't really want to do a simple random sample, because what if you take the simple random sample and you get nobody from a certain age group and all teenagers, which, may be unlikely, but it is nonetheless possible. And if we are interested in knowing how an effect changes on different age groups, we want to take a sample from every age group. So very good, everyone. I'm sorry, I only added that poll to the first class, to section 4. Okie doke. So these are the other two types of good sampling. The next thing I want to talk about is more about how you would design an experiment. So remember, we talked about two types of studies, experimental and observational. This section is called Principles of Experimental Design. And there are 3 of them. 1, control. The effects of lurking / confounding variables. So we talked about this a bit when we were speaking about observational studies, right? We wanted to create, in the case of observational studies, we had this example where people who wear a lot of sunscreen also have a more likely chance of having skin cancer. Whereas that doesn't necessarily mean that sunscreen causes skin cancer. Rather, it likely means that there is some sort of additional variable that is affecting both of them. So in the case of our stents example, which is sort of the example we've been coming back to over and over again with experimental studies, we have these two groups. We have a treatment and a control group. And these two groups are specifically for controlling these effects. Because we would say if there's any confounding variable affecting the treatment, then it should also be affecting the control. And the only thing that changes is this single thing. Now, there are 2 things within this control category that I want to talk about. 2 other things besides just splitting into treatment and control that we should think about whenever we're attempting to get rid of anything affecting the groups other than the treatment itself that we're trying to study. The first is something that I hope all of you have run into at least once before, or at least heard about, which is the placebo effect. This is when the knowledge that one is not In the treatment group, affects the outcome of the response variable. And the idea here is there is a difficult to quantify emotional effect to not receiving a treatment that we need to control for. Now, granted, it might actually be interesting to talk about and to study the emotional effect of receiving a treatment versus not receiving a treatment. That is, but usually whenever we do a study, we're trying to learn what is the effect of the drug even without this sort of effect. And I think it's usually the example you think about you think about whenever you think of a placebo effect is when the act of taking a pill actually affects your physical health. So if we had a study say we have a drug that supposedly removes acne. We give this drug to the treatment, give this drug to the treatment group, and give the control group a sugar pill. Until neither group which they got. This is because the act of taking a pill, whether or not it's actually supposed to do what we say it's supposed to do, can actually lead to changes, in this example, in acne, simply because of the emotional response of taking a pill. And this is a way that we do number one. We control for this effect by giving a placebo. In this case, the sugar pill is what we would call a placebo. Make sense? We go to this. I feel like this in particular is a concept a lot of people have heard about before because it's sort of, well, in my opinion, it's one of like the black magic things in statistics. Like, ooh, there's this that such a thing can actually affect you. I think it's gnarly. It's cool. Anyway, maybe that's just me. The other thing that is good for us to control, we do so using a double-blind study. And this is when neither the subjects nor the researchers know to which group the subjects belong. Treatment or control. And the example for this, and the thing we want to control for, is if a doctor knows one patient has the treatment, she may pay more attention or care to them than she would a patient in the control. So for each of these cases, not receiving a drug versus receiving a drug, having a care provider who pays more attention to you versus less attention to you, these can lead to emotional effects that may actually affect the outcome of the treatment. And we want to control for these, just like we want to control for bias and lurking variables. So these are all within this idea of control. And so it's not always feasible. Like in the case of our stents example, the patients probably knew whether or not they had received a stent. It might not actually, I doubt it would be feasible and perhaps not even humane to put all of them under anesthesia and after they wake up, don't tell them, open them all up, but only put stents into half of them. That sounds a little inhumane. But when you are able to, you should absolutely try to do all of these things. Okay. So that was the first principle of experimental design. The second one, randomize. Which is just use impersonal chance. To choose sample and to assign groups. If you do not randomize, you may introduce bias into your sample or into the groups that you decide to assign. And when you do that, the problem is that you may no longer be representing the population you are interested in describing, right? A sample is a representation of a population. If you have bias in your sampling methods or bias in the groups that you assign, you are suddenly no longer representing that population, you're representing something else. Maybe representing the population of people you would choose, right? And the third one is replicate. The treatment, replicate the treatment. Maybe I shouldn't have put a colon there. On enough subjects to reduce the chance variation in the results. So I'm going to post this again. I keep trying to take a photo. So remember when we actually went through and we calculated the proportion of stroke versus not stroke after a year? And we discovered there was an 8% decrease increase. My bad, I'm sorry. In the proportion of strokes. In the treatment group compared to the control. right? And I asked the question, is this 8% increase, is it true? Did the treatment truly make the chance of, did the treatment truly increase the chance of you getting strokes? Or was it just a consequence of random chance? And I further gave the example that if you flip a fair coin 10 times, we'd expect some variation in the amount of heads. We'd expect around 5, but we wouldn't necessarily expect it to be on 5. It would be completely reasonable to see 7 out of 10 got 5 or 4 out of 10 got 5, right? But if I were to flip it 1,000,000 times, I would expect it to be much closer to 0.5. So replicate this third principle, this third thing that we want to think about whenever we're designing experiments. It gets to a major statistical idea. And to understand this a bit, I'm going to define something that I hope is a word that you've all at least heard in some capacity, maybe by me actually. That is probability. The probability of an outcome is the proportion of times the outcome would occur if we observed the random process an infinite amount of times. So in the case of our sense example, what are we actually interested in quantifying? We want to know that the probability of getting a stroke has gone, hopefully, down whenever we give people stents, right? But note how we define probability. Probability is a proportion. It's just the proportion of times it happened if we did it an infinite amount of time. If over the course of human history, we gave everyone with a risk of stroke a stent and recorded how many times the person with the stent got a stroke and didn't get a stroke, right? Of course, that's infeasible to do. Taking an infinite amount of samples in any regard is not humanly possible. So what we did, of course, is what we always do. We take a sample and we calculate the proportion of the sample. But note the definition of a probability. The probability is itself something, is it a proportion, excuse me, I'm not explaining this very well. The fundamental idea here is that we are going to get closer and closer to the actual probability value the more samples that we take. And that's called the law of large numbers. And the idea here is as more observations are collected, the proportion of occurrences in your sample converges to the probability of the outcome. So if we could know the probability of getting a stroke, and we could know further that stents either increase or decrease that probability, we would be very happy people, right? That would solve our problems and we would know whether or not we should be giving people stents. The thing here is, of course, is that probability is not something that we can really explicitly calculate in this case. So we took a sample. And the point I'm trying to make, because of this last little thing I've mentioned here, because of this law of large numbers, the more observations you take, the closer and closer you are going to get to this true probability. It's going to approach the probability. There may be some variation, but it's going to get closer the more samples you take. So the final point of these principles of experimental design is to take as many samples as you can. The more the merrier, all right? I'm trying to think if there is an example of a situation where you wouldn't want more samples. If there is, I assure you it's very fringe and it's outside of the scope of this class. So principles of experimental design, any questions? All right, moving right along to mosaic plots. Oh, this is fun. I like mosaic plots. I think they look weird. Okay. So let us recall our email data. This one right here. So remember we had 50 different observations of emails, all given different classifications like spam or not spam, and we have also recorded numeric variables on them like number of characters and number of line breaks. So we can draw the following plot from our data, from this email data. Let's start with this one. I'm going to call it my first mosaic plot. Cool. So how do I read this? Start by just looking to understand what this plot is describing. Start by just looking at the top bar. We have our three categories within the number variable. So this is this first variable right here, number, where the categories were none, small, or big. If you look at the width, the horizontal width of each of these boxes, so I am talking about, oh, and my colors are kind of bad here. I'm talking about this width right here, this width, and this width. That is the proportion of the data that belong into each of these three categories. So just as a guess, I might say that this is maybe around 0.1, maybe this is around 0.8, maybe this is around 0.1. So without even thinking about what's happening on the horizontal bar on the left side, the top tells you, well, of all of the emails, 10% of them were classified into none, approximately 10%. Approximately 80% of them were classified as small, and approximately 10% of them were classified as big. Now, once you understand that, the horizontal side, the left side, spam or not spam, tells you within each of these categories what was the proportion of spam and not spam. So let's just look at none for now. What this is saying is within the none category, there were this proportion of spam and this proportion of not spam. Now that is not saying of all, so maybe this is around here. Maybe I'll do the same thing here. This looks like it's around 0.3 maybe, and maybe this is then 0.7. This is not saying that 30% of the emails were spam and 70% were not spam. It's saying if you look at just the emails that are classified as none, 30% of those emails are spam and 70% are not spam. Does this make sense? All right. And you would go further to the, as you read left to right, you would do the same thing for the other categories. Within the small category, it would appear maybe like, oh, I don't know, like 95% were classified as not spam and only 5% were classified as spam. Now, something to note here is that it absolutely does matter in what order you put the variables, whether it be, in fact, why don't I just show you? So notice that this mosaic plot is the other way around. Spam versus not spam is on the top, and on the left side, we have the different numbers, none, small, or big. And in this case, spam or not spam, this is referring to the whole data set. In the whole data set, this proportion, it would appear were not spam, and this proportion was spam. And then considering now only the not spam category, this amount in the not spam category was none, this amount was small, and this amount was big. Any questions on this? I think these plots, like when I first saw them, it was like, oh, I don't know what this is saying at all. And then when it clicks, it shows you the relationship between two categorical variables, which complements what we were doing last time. We were using scatter plots to look at the relationship between two numeric variables.
Does it just flip based on kind of what you're like trying to like graph from the data?
So let me answer that how I comprehended it, and let me know if I misunderstood your question. Really, okay, so they are two distinct graphs, and they're not just each other flipped, rotated 90 degrees. They are actually very different from each other. And you would choose the order in which you want to graph them based usually on what question you're asking, right? This plot right here answers the question, what is the breakdown in each of the number groups of spam and not spam, right? Whereas this is saying, what is the breakdown in each of the spam groups of number? Maybe that's a strange way to say it, and perhaps we should try to motivate it with a more reasoned research question. So how about this? Empirically, if I knew which number category I was in, would that affect how likely I would expect to get spam? And what I would read from this plot is, well, if my number is none, the proportion of spam in the none category is bigger than the proportion in any other category. So if you were to give me a number, none, small, or big, and you were to say, in which one would I expect to have spam the most? I would say, well, none, because I've observed that within the none category, spam is the highest. The proportion of spam is the highest. Does that answer your question?
Yes.
Awesome. Well, I guess it all comes down, a lot of these visualizations, it comes down to what you're interested in, which is that first point of the statistical process, like develop a question, right? Which I mentioned I don't really go into as much because what you want to know is ultimately up to you. I just want to give you the tools to be able to figure it out. All right. So those are looking at the relationships between two variables. The last thing I want to go over today is talk about visualizing single variables. Oh, okay, so I want to go back to my cold color. Wow, I made this header so big. So, still working with our emails data. Let's look at just numbers. So that's the one that's none smaller big. Here are two examples of single variable charts. This one is called a pie chart. And this one is called a bar chart. Now, note, these are really giving us the same information, but I will give us a bit of a warning. The y-axis here is frequency. So what this is saying is there were, what, a little over 500 in the none category, a little over 500 in the big, and then a lot in the small, right? And then what here we have are proportions. So this is the proportion of the circle that were none. If I were to just, add up all of these and divide by the total to get the proportion that belong in each category. This is the proportion that belong in big. And then all of the blue here is the proportion that belong in small. So this gives us an idea of the breakdown of a single variable. And you'll see actually, if we look back up here, it matches just the top bar of this plot right here, right? A little bit in none, a little bit in big, and a lot in small. Same thing here. A little bit in none, a little bit in big, a lot in small. I will say there was something that the book mentioned, and that is they don't like pie charts. I don't really have a strong opinion on pie charts versus bar charts. What the book reasoned was that bar charts better visualize and give you more information than a pie chart does. I like a little bit agree with them, but ultimately I would just, you should do what you think is most informative whenever you're trying to present data or whenever you're trying to understand data for yourself. I mention it only because the people who wrote this book are very excellent statisticians, so perhaps their opinion should hold weight for you. Also, I'm not going to spend too much time on these two particular plots because I find them rather intuitive, and if you don't, you should reach out. We should talk about them during office hours. The next type of single variable chart we're going to do is for a single numeric variable, and it is called a histogram, and that's what your next video lecture is on. So that's all we're going to talk about today, actually, as far as new material. As soon as we start going through histograms, we're going to get to move toward more numeric calculations and explicitly quantifying things like probabilities and how likely certain events are. Are there any questions up until now? All right. If there are no questions, then the video is posted. You may go ahead and get started.